Welcome everyone to our ECSS Symposium. So it's my pleasure to host this meeting today. Bob Sinkovitz sends his regrets. He really wanted to be here and had a conflict that came up suddenly that he had to attend to. So he asked for me to fill in for him. So I'm Phil Blood. I'm with Bob, co-director of the ECSS program in XSEDE. And so today we'll have the opportunity to hear from David, Mona, and Jeff. David's coming to us from UCSD, and then Mona and Jeff from San Diego Supercomputer Center. And together they'll be talking about high-resolution spatial temporal analysis of whole-head 306-channel magnetoencephalography. Oh man, I knew I was going to mess that up. Magnetoencephalography and 66-channel electroencephalography. No, brain imaging in humans during sleep. And so one thing I will expect is for our speakers to roll those things right off their tongues without any mangling like I just did. So I will, without further ado, hand it over to our speakers.
Thank you, Phil.
And actually, can someone give me a host presentation ability, please? My internet cut out and I had to come back in.
I don't see anything about our slides on the screen.
Yeah, so let's see. Marcus, do you control the ability to share here?
All right, there we go.
There we go.
Yeah, it's open for anyone to start sharing now.
All right, my name is David Shannahoff-Khalsa. I'm from the Biocircuits Institute in the Center for Integrative Medicine at UCSD. And since I'm the first speaker, I want to do what normally we do at the end of a talk, and that's give acknowledgements. And I want to start by acknowledging Mona, who founded me and asked me if I wanted to collaborate on a project. And when I found out she was at the Supercomputer Center, I realized, yeah, wow, what a blessing this is, because for 8 years I'd been looking for somebody that would have the time and the interest to work on this data set, which was at least at the time that Mona and I met, a one-of-a-kind sleep data set in the whole world. And you'll see why as we go through this. But so then Jeff came on board a couple of years later, and he is also an amazing talent, both as a scientist and as a visualization expert, or artist, as I'd like to describe him in that way. But next slide, Mona, please. All right, we also have other collaborators. One is Shah Gulshan. He's from the Department of Psychiatry. He's a biostats expert. Nima Degani from MIT. Eric Hallgren from the Departments of Radiology and Neuroscience at UCSD. Eugene Zawadsky, who's from a private company in San Diego, who's collaborated with me on this project, this kind of project in the past, and Arlene Schlosser, who is now retired, but she was in the Department of Psychiatry and did all the polysomnography. She was a sleep expert and probably still is, and probably was the most talented in the county at the time that she did our sleep hypnograms. Next slide, please. So all of us have probably our most popular slide ever, and I want to give you some background now and what this project is all about from a biological level or in terms of neurosciences. There's a paper that was published in Human Neurobiology in 1983. It was entitled Alternating Cerebral Hemispheric Activity and the Lateralization of Autonomic Nervous Function. And what that essentially means is that the two hemispheres of our brain alternate in dominance with an ultradian periodicity. And they alternate in dominance during both waking and sleep. And they're coupled to other systems, particularly the autonomic nervous system. We've all had a common cold and found that one nostril can be completely congested for a couple hours or something like that, a fairly long time. And then if we're really paying attention, the next time we check, it's the other side. Well, that's called the nasal cycle. It's a natural endogenous autonomic rhythm that actually is a marker for for how the two hemispheres of the brain are alternating in dominance. And we published that, like I said, in 1983. It was done in the Department of Neurosciences here. And what we have on this slide is 3 subjects out of, I think, about 22 that were included in this study. And the top line says airflow, for GF is the initials of that subject, shows above a line, below the line, above a line, below the line. That's right nostril, left nostril dominance. And you can see a time marker down towards the bottom right at 15 minutes. We kept these people in the chair, in a relaxing chair, and asked them to do nothing, and we were just watching their natural endogenous rhythm. And below the airflow, you see total EEG, alpha, theta, delta, and beta, and you can see right offhand that they all sort of parallel each other very nicely. Subject PC below also shows, and it shows that the right nostril and left hemisphere are dominant for total power. And then JW, the last subject on this in this figure, switched actually about every 25 minutes. We looked at her on another day, and she was more similar to the upper 2 subjects. So the ultradian rhythms, especially of the brain and anything else in the body, to be clear, has a great deal of wobble, meaning it's the variance of it, and it can be huge during the day. So next slide, please. This is a slide that's measuring catecholamine, norepinephrine, in the peripheral circulation. We have 2 indwelling catheters, one in the right arm, which is marked by the red line, and one in the left arm, and with the green hatched line. So every 7.5 minutes, we're sampling from both arms, and we're plotting the values in the upper part of the graph. And then we do left minus right. We get the graph below, and you see this nice ultradian parameter. So everything on the 2 sides of the body is out of phase as well, at least during resting state, which would include sleeping, of course. Next slide. That work was also done at UCSD and was published in Life Sciences in 1986. This is a study that was published in 2000 in an International Journal of Neuroscience, titled Ultradian Sleep Rhythms of Lateral EEG, Autonomic, and Cardiovascular Activity Are Coupled in Humans. Now, what I did here is that we plotted the nasal cycle, the top part, inversely compared to before. But if you were to look at it closely, you'd see that these things look nearly identical, and they're inverse coupling once you start to study the picture in any detail. So this was the first time that we showed the nasal cycle and EEG alternating patterns in humans. We were also the first to show it in the waking state. Next slide, please. And I want to read to you something that we had in a paper that's going to come up in a moment. It was entitled Ultradian Rhythms of Alternating Cerebral Hemispheric EEG Dominance Are Coupled to Rapid Eye Movement and Non-Rapid Eye Movement Stage 4 Sleep in Humans. That was published in 2001 in Sleep Medicine. And I want to read the first paragraph so you get a better sense of what we're going to be looking at in our data. In 1972, Goldstein et al. Measured EEG with the intent to look for hemispheric asymmetries coupled to rapid eye movement and non-rapid eye movement sleep cycles. Their results in 7 humans showed that left hemisphere EEG power dominated during REM sleep and that right hemisphere EEG dominated during non-REM sleep. They found the same relationship in 3 of 4 cats and 3 of 5 rabbits, with 1 cat and 2 rabbits showing opposite coupling. They concluded that the difference in hemispheric amplitude relationships during non-REM and REM sleep may eventually prove to be a neurophysiological concomitant of the changes in brain function during sleep stages. So that was published in 1972 in Physiological Behavior. You can see the reference below. Next slide, please. So this is the data. This is 10 plots of 10 young, normal, healthy males that had relatively very little muscle artifact that would otherwise interfere with the EEG signal in our recordings. And you can see there's a period-like phenomena going on here, where it's going above and below the midline and so forth. The third one down on the left, of course, looks more sinusoidal, and that's what you expect in rhythms when you think about them. But in reality, these kinds of ultradians look more like this. So that's just 10 young, normal, healthy males. I can't remember whether they were all right-handed or not, but after doing that, there were 10 of them that followed exactly what the majority of the Goldstein et al. People found, without any exceptions. But we did find an exception. Next slide, please. And before I describe this slide, when I saw the exception in our thing, I thought, okay, maybe Mona made a mistake in analyzing the data, but we'll see that downstream. But of course she didn't, because she's nearly perfect at everything that she does, and she's amazing. And so on the right side here, what we have is just a single hemisphere of smooth data with delta, theta, alpha, and beta. And as you can see, the sleep hypnogram above, where it's 4 at the top of that stage 4, and R at the bottom is REM. So those are the polar opposites for sleep states. Now on the left side, we have the same hypnogram, of course, but we have the left minus right patterns. So you can see that during the stage 4, the first stage 4, we get a big dip into the right hemisphere. And then it comes back up for REM. So we did statistics on all of that. And at least with delta, and I think one of the others, I don't remember which other frequency band was showing the significance. But next slide, please. All of this is described in a book that I was invited to write for a review series called International Review of Neurobiology. It was volume 80 in 2000, it was published in 2008. I have no financial benefits from this book, so there's no conflicts of financial interest. And it's entitled Psychophysiological States, the Ultradian Dynamics of Mind-Body Interactions, published by Academic Press and Elsevier Publications. So I was very happy to have that invite and very proud of having all of my hard science published in this book. But the stuff we're going to see today is not yet in the, well, it's not in this book, and it won't be until if there's ever a revision. But I just wanted you to see that there's this comprehensive analysis of how all the ultradians, even calcium in your blood, hallucinations, all of these sorts of things have ultradian periods. And so the alternating dominance of the two hemispheres is the primary control factor. And actually those hemispheres are controlled by the hypothalamus, which controls all of the other ultradians in the body through the autonomic nervous system. Next slide, please. So here's what the MEG, magnetoencephalography instrument, looks like. And one of the reasons that we probably have the only long 4 to 6 hour recordings of sleep with subjects is because you can imagine trying to sleep in that without turning your head. If you're a back sleeper, you'll probably be okay. But this instrument is about a $2 million instrument. And part of the challenge is not only is it expensive to buy, but then you have to build a special room for it so the external fields from the planet don't interfere. And this makes it a challenge, and then you have to have helium to run it. So unless you have helium close by, you're not gonna be able to run an instrument like this. So they're very rare instruments. Next slide, please. This is what the head array looks like. Each one of those purple little things is like 3 channels. There's 102 of these on the head. There's 1 magnetometer and 2 gradiometers at each site. So we have really 306 different channels with 102 places on the brain. Next slide. That's what it looks like in a two-dimensional. So we have frontal, parietal, temporal, and occipital regions. Next slide. That's the EEGs. Those signals are more common, of course, and people sleep with EEG electrodes on their head all of the time, but it was added to this instrument to give people 4 different signals to look at. Next slide. So Mona took the raw data, which she'll explain a little bit about, and then she converted to low delta. She did a frequency band analysis, and we looked at low delta, delta, theta, alpha, beta, and gamma, and you can see the frequency of each on the right. Those were suggested by Eric Hallgren, our chief brain in sleep and MEG. Next slide, please. Next slide.
You're not seeing it?
Hello, Mona.
Yeah, can you hear me?
I can hear you, but the slide's not changing.
Oh, my internet is unstable. Rats. I have changed it. Hopefully it'll come through.
It hasn't yet. So I don't know what slide we're on. Okay, that's those are the 4 hypnograms. They're plotted somewhat inversely compared to the sleep medicine paper, but you can see that they look very, very different. At the top of each is for waking. And then it's REM sleep, and then stage 1, stage 2, stage 3, and stage 4. This is a more common way that the hypnograms are plotted these days. But subject 1 has a near ideal hypnogram where stage 4 sleep shows up almost immediately as the subject went to sleep. Sleeping in the MEG instrument is not easy, and that's probably why subject 2 on the bottom left showed almost no stage 3 or 4. And subject 3 upper right had a lot of waking, but it also showed the early onset of stage 4. And then subject 4 only had stage 4 at the very end of the night, which is unusual, but apparently he finally, or she finally went into a very deep state of sleep towards the end, out of necessity, of course. All right, next slide, please. All right, this is just an example of left minus right, low delta, delta, and theta, how these things vary. And on the right side, we have alpha, beta, and gamma. And this is before we detrend the cycle. Next slide, please. This is the signal detrended, and you can see that there's not the same kind. I mean, it looks the same, but we detrended the signal so that we wouldn't pick up, 6 or 7 or 8 hour rhythms if it should be the case. So we're only looking at cycles up to 5 hours with 5-minute intervals. All right, next slide, please. Next slide.
I did advance. Let me try again.
I'm still just seeing the detrended
Wow, my
figure.
internet, it must be really poor. I am wired.
I'm not sure I can promise my internet's going to be any better, but I would be
Right.
happy to do the sharing if you think.
This is now the FOS plots. Fast orthogonal search is like an FFT, but it's more sensitive to nonlinear dynamical data, which all human data really, or any physiological data is always nonlinear. And what we have is subject 1 in all 4 of those plots, but we're looking at the magnetometer in the upper left, the EEG on the bottom left, and the G2 and G3, the two gradiometers on the right. They look similar in some ways, but there's also differences. And so that's what, that's how much sensitivity these signals, how they can differ from one to the next, even though supposedly we're recording the same thing. We're close to the same thing. Next slide, please. This is comparing whole head at the top, the back half of the head in the middle, and the front half of the head in the on the bottom. So even with one type, i.e. the magnetometer, you know, with the different regions of the brain, we show these kinds of variations. Next slide, please. This is again the magnetometer in 4 different FOS plots, looking at frontal, occipital, parietal, and temporal regions. So that's about, you know, almost an equal amount of signals for each region. So again, it's showing us that there are similarities, but there are also differences if we're looking at just different regions of the brain all at the same time. Next slide, please. This is, we have been selected, and I don't want to go into this in too much depth because we're running out of time, I am. But we're going to be looking for bins from 20 to 65 minutes, 70 to 100, 105 to 150, 155 to 205, 210 to 265, and 270 to 300, which is pretty similar to what we've seen in past studies. And instead of boring you with this, more of this slide, let's go on to the next one. All right, and this is where Mona begins. So I'll leave it to Mona. Thank you. Questions, you can ask questions later if you want.
Thank you, David. I also want to express my thanks to David for his openness for our collaboration and for his guidance and patience as I learn MATLAB and also these data processing steps. I want to talk a little bit about the data. The original data was in a proprietary NeuroMag file format. We had 4 subjects, 3 females, 1 male, and their age range from 20 to 30. And all the subject data are recorded at approximately 603 hertz with a total of 389 channels, which contain 4 main types of signals, which David, I think, had mentioned, the EEG, the 2 gradiometers, and the magnetometer. And this gave us a total of 366 data channels per subject, and the remaining 23 channels we didn't use. Here are some of the sort of resources and tools that we used. We processed most of our data on SDSC's Comet in interactive mode. I think everyone knows MATLAB, and we use that for most of our data processing. Field Trip is a MATLAB toolbox. It's an open source, and it's meant for EEG, MEG, and 2 other modality analysis. And we used it mainly to read the proprietary NeuroMag files. OSF is an open platform that supports research and collaboration from the Center for Open Science. And our latest tool is XSEDE, a data sharing resource for the SDSC HPC users. We wrote custom MATLAB code to do most of the processing steps. First, the NeuroMag files are stored in separate, what they call trial files that end in a .fif. And so we converted the proprietary data files into a MATLAB format using that Field Trip toolbox. And as you can see from the table, each subject had over a dozen data files, which when stitched together, gave us millions of recorded points over several hours. And for subject 3, we did have a corrupt file, interestingly enough, file 13. And on the left, you can see an example of the combined continuous data for subject 3 for one of the channels, channel 20. And as you can kind of see, I don't know if you can see my mouse, but the gap in the middle there is the 13.5 minute data gap from the correct file. And we originally padded it that with 0. However, this zero padding didn't work very well for later analysis. And you can see an example of that on the upper right, which produced a large spike artifact. So we tried a few things to attempt to smooth out this large artifact, and MATLAB's fill gap function worked well. It uses a forward and reverse autoregressive fit, and the right side bottom shows the results of the same data using this fill gap. Next, we separated out the 4 signal types of interest, and then we performed the bandpass filtering from the 6 bands that David showed earlier. And as you can see, even at this early step, we already had nearly 42,000 pieces of data. Additionally, we had hundreds of files as we saved the process results in both MATLAB and ASCII format. And then starting at the RMS step, we also plotted all the results. So each subsequent steps produced about the same number of outputs, so data and file management strategies were very important. And for the RMS, the calculations were done using non-overlapping windows of 603 points, which is approximately 1 second. And this reduces the number of data points, but as you can see from the plot on the lower right, there are huge noise spikes in the data, which make the signal, which is at the very bottom of near zero, very difficult to see. So we tried various ways to denoise the data. The last 2 in bold showed what works well. And as you can see, an example of the before and after clipping on the plot to the right. We also tried various MATLAB functions, including filter, both forward and backward, and shifting. Also the fill function, which is a 0-phase filter. But the 2 that produced, I think, the result that David felt more comfortable were the Hampel and the move mean filters. And for the Hampel, we used 100 neighbors on each side, and the bottom plot shows an example of before and after the Hampel filter. Notice that it didn't get rid of like fat spike noises, which you can kind of see about to the right of the plots. And then for the move mean, we used a sliding window of 499 values. And earlier, David has shown pictures of the MEG channels. Basically, homologous pairs are channels that are considered to be the same position on the right and left side of the brain. So the image on the upper right shows a few of these pairs in colors for the MEG. And because our project is interested in the hemispheric dominance, we calculated the individual left minus right homologous pairs. And the bottom plot shows an example of one such pair for subject 1, channel 55 in blue, and channel 99 in red, and the results of their subtraction in yellow. We removed a few channel pairs due to bad data. This was a kind of a manual process and with several iterations. Later, Jeff's visualization was very helpful to identify more bad channels that we had missed during our manual review. And the table on the right shows the percent of bad channels that were removed from the final left minus right calculations. And then lastly, we detrended all data. I think David has showed a couple of pictures earlier. Here, I show an example from subject 1, where the blue is the original left minus right mean, yellow is the trend line, and orange is the new detrended data. And we're also very lucky to have Jeff join.
Hello.
Yeah, I'm not sure. Did we lose you, Mona? Well, I suppose the timing might be good. You know, we still see your slide, but I think this is where I jump in.
Yeah, go ahead, Jeff.
Okay. Oh yeah, I'll need you to stop sharing, I guess.
I did. Gosh, my
Okay.
internet is
Yes.
terrible.
No, no, we're good. Oh, I'm sorry, I actually didn't pick the right thing. Let's see, there we go. Okay, can everyone see my screen?
Yes.
And you can't see my notes, hopefully. Okay, so thanks very much, everyone. I also want to thank XSEDE ECSS and ESSGW, and I also would really like to thank David and Mona for the opportunity to work with them and with this data, and for being so supportive of my somewhat unconventional visualization methods. Okay, so to begin, the slide that we're looking at right now is 4 different combination bar plots. And the focus of these slides is to identify the activity in stage 4 in particular for the lower frequencies. So each one of these plots is a combination of 6 bar charts, and the individual bar charts are color-coded based on the 6 different frequencies, with low delta and delta being blue and orange respectively. And the thing we're really focusing on is stage 4, highlighted in the purple rectangles here. And we can see that the low delta and delta bars are, in case of low delta, negative, which is consistent with the hypothesis that lower frequencies are more dominant on the right, in the right hemisphere in stage 4. Now, there were some exceptions to this, and this slide briefly reviews some of those. In the case of the whole head data, there were 1 out of 24 opposite to the hypothesis. For the front and back regions, 5 out of 48 were opposite to the hypothesis, and in the 4 regions, only 16 out of 96 were opposite to the hypothesis. But the focus of my presentation is on visualizing spatio-temporal data sets and trying to visualize the entire data set at a single time. And I'll be reviewing some list of methods that are listed here, ultimately focusing on 3-dimensional spatio-temporal isosurfaces like the one that's shown on the right. And I'll go into greater detail as to what those are in the next few slides. But before we get to that, I want to mention that this project presented us with some challenges. As Mona mentioned, thousands of lines of custom MATLAB and Python code were written to generate some of these visualizations because there was so much data and it was so extensively processed. And also because some visualization methods were relatively unconventional. They aren't default functions in visualization tools. And we also faced challenges with XSEDE systems which weren't able to support OpenGL hardware acceleration in MATLAB. And that significantly slowed the production of our visualizations and animations, which forced some workarounds, but we were able to work around those. So next, I want to briefly review the nature of this data. First, the spatial nature of it. This is an image you've already seen of the MEG and the EEG channel layouts on the scalp as viewed from above. They have 2 spatial components, the X and Y components of the channel locations on the scalp, and a temporal component of 1 sample per second after the data was reduced. And I want to point out also the front, back, left, and right locations of the brain are identified. And it's important to keep those in mind, those viewing directions, because they'll be important when we look at the more advanced visualizations. So first, let's just look at the temporal component of our data set. How can we visualize the entire data set in a meaningful way? Well, maybe the simplest way is to plot all 102 channels as a time series in a single plot and use color to differentiate one channel from another, as shown here for subject 1's G2 delta activity. However, with 102 channels, the legend runs off the screen, as you can see, and the colors are duplicated, making it difficult to identify any particular channel. Also in this data, there are 3 bad channels, as Mona mentioned, which are not visible because they're actually flatlined at the 0 value of this plot. So one way to resolve some of these problems might be to use what's called a cascade plot, which is used in brain data visualization. And here's a screen grab from EEG Lab, which is one of the most popular open source EEG analysis tools, also developed at UCSD. The data set we see here from EEG Lab tutorials is only 32 channels, and even though we can more clearly make out individual channel dynamics, it's not clear where the channels are located on the scalp. And there's no easy way to apply a vertical color bar or vertical axis labels to identify values of power for the brain activity. But here's an example of doing something similar with our data, and we can more easily identify some of the bad channels highlighted with red rectangles. But with so many channels, we still can't easily identify the spatial locations on the scalp to help gain insight into hemispheric specific activity. So another approach might be to use a two-dimensional contour plot or a two-dimensional heat map. One advantage of these methods is the use of color to represent values of power. So now we have a color legend, and we can also see the 3 bad channels identified here by red arrows. However, there's once again still no way of easily identifying spatial locations of the channels. But one interesting thing about this method, at least in MATLAB, is that our heat map is also a 3-dimensional mesh, and we can rotate it and tilt it to a bird's eye view. And this is equivalent to merging the time series plots with the heat map. In other words, the vertical peaks are analogous to the time series we viewed earlier. So we're also using color and elevation to represent values of MEG power. And if we include the subject's hypnogram, we can make visual correlations between brain activity and sleep stages. For example, highlighted in the purple rectangles, we can see similarities in brain activity for stage 4 sleep at different times during the night. But we're still uncertain about the spatial locations of the channels. Okay, so let's discuss the spatial components of our data and introduce two-dimensional topographic brain maps. 2D brain maps have been used for decades in brain data visualization, and this is another example from EEG Lab, where we see 3 circular colored plots, and these are the 2D topo maps. Each one has a black circle overlaid with a little point at the top for the nose and 2 boxes left and right for the ears, as though we're looking at the brain from the top down. And the bottom is the back of the brain, or what we'd call the occipital region of the brain, consisting primarily of the visual cortex. And highlighted in the red arrow shows a red region in the back of the brain, or the occipital area, which probably corresponds to the classic 10 Hertz alpha activity, suggesting that the subject was probably relaxed with their eyes closed. Once again, this is not our data. That example was not from our data set. But this example is, and this is intended to give you an idea of how 2D topographic brain maps can be used in animation. So let me go ahead and play this. And I don't expect it to play well, and I don't intend it to actually animate. So I'm going to pause it and then jump forward a little bit. Above, you can see the 2-dimensional brain maps. We generated these using Python code because we were working outside of EEG Lab. In the middle, the red plot is the sleep hypnogram, and at the bottom is one of the time series from subject 1's G2 delta activity. But if we step through the data, we can see the lines line up with the different sleep stages, and we can draw visual correlations with the locations in the 2 different hemispheres and where we see high activity in red and low activity in blue. But one of the, so one of the advantages is to see the spatial distribution, but it's at the expense of the temporal component, meaning as we scan the video back and forth, we are forcing our brain to remember what we were looking at as we jump around. And so one way to resolve this is by using what's called the principle of small multiples, shown here. So this is a sequence of 8 2D brain topo maps at 10-minute intervals, and this allows us to easily scan the time-stamped image sequences to make correlations and see trends more clearly. And the more the maps, the merrier, I guess, but at some point we run out of space because it can be prohibitively difficult to identify trends or anomalies with too many maps. So, if only there were a way to combine both spatial and temporal components into a single visualization. Well, here's an approach I used almost 30 years ago. Imagine if we ordered the data set as if we were stacking the sequence of 2D brain maps like a loaf of bread or a stack of poker chips, where time is vertical and earlier time is at the bottom and later time is at the top. And to be very explicit, what we're looking at here is, so we can orient the time series themselves vertically and arrange them spatially so their positions correspond to their location on the scalp. I don't mean to imply that this is a practical visualization. It's just meant to orient you as viewers to what we're actually looking at here. But here is something that's perhaps more of a useful approach, using what might be called a 3-dimensional stacked scatter plot, in which we use a stack of scaled and colored ellipses to represent brain activity. And here's a closer look. And this method is actually gaining popularity in general visualization applications, particularly GIS visualization. One of the problems with applying this method to our data is that the data is compact and the outer channels can obscure the activity of the inner channels. Perhaps geospatial data sets are more conducive to this kind of visualization, such as the one that I'm showing here. This is a visualization I did of the COVID-19 daily deaths a couple of weeks ago over the last several months of data. And the distributions of the ellipsoids are looser and a little bit more widespread, so nothing is too badly obscured. Time in this case is also vertical, and we can see the map below to make visual correlations with the geospatial locations. So my conclusion was that this is a great job for spatio-temporal isosurfaces. And the images that you're seeing here are almost 30 years old when I had access to the 3D visualization application AVS, which is still around. And this was one of the first applications of this method, to my knowledge, on brain data or any data for that matter. And this is mainly EEG data we're visualizing here, except for the one in the lower right, which is heart data or ECG. And I wish I had more time to talk about what these actually are, but I will be explaining how they were created and show some examples of applying them to our own sleep EEG and MEG data in this data set. Okay, so first, what is an isosurface? Many of you probably already know, but for those who don't, an isosurface, a 3D isosurface, is a three-dimensional analog of a two-dimensional isoline or contour line, such as the ones shown here, which are typically found in 2D topographic maps used in orienteering. And isosurfaces are definitely a useful, well-known visualization tool found in most 3D viz packages, including MATLAB or Visit or ParaView. And for decades, since their inception, the convention has been to visualize objects consisting of three spatial dimensions, like anatomical structures, such as the arteries in the upper right, shown here, or fluid dynamics in the lower right. I think the assumption is that the object exists spatially. It has three spatial dimensions, good old X, Y, and Z, independent of time. But this is just a convention, not a required constraint. In fact, this is still the definition found in Wikipedia. But in fact, isosurfaces don't require that all three dimensions be spatial. In fact, none of them actually have to be spatial. So here's a slide, hopefully to help explain the idea behind 3D spatio-temporal isosurfaces. So the image on the left in step one shows a vertical three-dimensional box-shaped volume, and you'll see at the very bottom the MEG channel layout aligned horizontally like a slice of the three-dimensional volume. Time's vertical, earlier time's at the bottom and later time is at the top. And then we stack the 2D topographic brain maps like a loaf of bread as shown in step 3. And in step 3, I've also included a single isosurface colored in red to show regions of high activity, but that's just one single isosurface. And in the rightmost image for step 4, I've added 20 individual isosurfaces to fill the volume and show areas where there's low activity in blue and medium activity in the yellowish and greenish or the cyan areas. Okay, maybe to give you a better idea of what it is we're looking at, here's a movie that I wanted to play so you can get an idea of how MATLAB makes it possible to work with and interact with spatio-temporal isosurfaces. So here I've stacked 6 different collections of isosurfaces, and each of the 6 towers represents brain activity for each of the 6 frequency bands. And you can see that's labeled near the top there. On the far right is the sleep hypnogram, and it's oriented vertically because I've got time oriented vertically. And as we step through this animation, we can see the isosurface towers are rotating. So we can view them from any different angle. And this is all something that MATLAB is a really nice interface with MATLAB. Visit has, similar types of interactive interfaces as well. And that's pretty typical for these kinds of isosurface visualizations. And I also want to mention or point out that these 3D volumes can be sliced vertically, and these vertical image slices, which are vertical equivalents to the horizontal brain map slices, they can be animated too. We haven't had the time to get to that, but I'm excited to try this application because I think it has a lot of potential for revealing deep structures. Okay, so now we're going to review some simple examples of how this method can make it a bit easier to observe interesting structures. So here we have subject 1, MEG channel M1, viewed from the right. The 6 isosurface structures shown here correspond to MEG brain activity. And on the far left is low delta, and then theta and alpha, beta, gamma as we go further right. One interesting feature of just about all of these visualizations is the similarity between neighboring frequency bands and the significant differences between the low and the high frequencies. And subject 1 is a good example of this. And by this I mean low delta is very similar to delta but not identical. Delta is similar to theta but not identical, and so on, such that there appears to be sort of a morphological gradient from left to right, which I find really interesting and compelling. And I think it helps validate all of the monumental processing that Mona did leading up to this. There were so many steps, as you've seen in the processing workflow, where Murphy could have reared his ugly head, but it looks great. And so as we saw earlier with the 2D heat map, one of the most obvious features for subject 1 are the 3 occurrences of stage 4 and 2 of stage 3 as highlighted with these purple rectangles. And here we observe similar brain activity in those regions across all frequency bands. Now, in some of David's and others' previous studies, and as has been mentioned already, for many subjects, a general trend is observed in which the left hemisphere is dominant during REM sleep and the right hemisphere is dominant during non-REM stage 3 and stage 4 sleep. And one of the primary goals of creating these visualizations is to help identify the extent to which that trend is valid, at least in our data. And I'll talk more about that in just a little while. Here we have subject 2's MEG channel G3 viewed from the left side of the brain. And in this visualization, we can see highlighted in the purple rectangles that the higher frequencies, beta in particular, appear to be somewhat of a mirror image of the lower frequencies. And by that I mean what's blue in beta appears to be red in low delta, and what's red in beta appears to be blue in low delta, and so on. Here in subject 3's MEG channel 1 viewed from the right, we see on the far right in the earliest waking state for gamma, it's distinctly different from the later waking state nearer the top. And to some extent in beta as well. And we don't observe this in other frequency bands. Here we have subject 4, MEG channel 2, viewed from the front of the brain. And in this visualization, we see similarities between brain activity during the 2 occurrences of stage 4 sleep. And also beta, again, appears to mirror the lower frequencies. This is a visualization of subject 1, EEG, viewed from the front of the brain. And one thing to observe is the high beta activity during REM, which might be expected since we are dreaming during REM and our higher cognitive functions are especially active. And on this slide, highlighted in the rectangle, alpha and beta appear to mirror each other, especially during the middle part of the night's sleep. And we didn't observe this with other subjects. So this is really interesting. Often alpha and beta will look a lot more similar. But we can see, especially during the middle part of the night's sleep, what's blue in beta is reddish in alpha and vice versa. There's a lot more going on in these data sets beyond that. And finally, in this slide, we see high beta and gamma activity during the deeper non-REM sleep stages, highlighted with the rectangles. And this might be unexpected. Typically, in the deeper sleep stages, the higher cognitive functions are not active. So I'm not sure what the explanation might be there. Okay, but one of our main focuses is the relative dominance of the left versus the right hemisphere, and that was the reason for creating the left minus right homologous pairs. So this slide reviews that process that Mona had already explained. And in the lower right is a schematic of constructing a 6x10 grid with the remaining 48 pairs of homologous pairs. And this 6x10 grid can also serve as a cross-section for 3D spatio-temporal isosurfaces. So here's one example. And one of the most interesting things about these versions is that whatever is red, whatever is higher values in power, implies that the left hemisphere was more dominant or more active than the right hemisphere for that particular region. So it makes it real easy not to even have to worry about whether you're looking at the left or the right side of the brain. So here highlighted in green, in purple, we see the right hemisphere is dominant during the earlier stage 4 events near the bottom. However, in the later stage 4 and even the stage 3 event, we see significant left hemisphere dominant activity. That's not to say that the right hemisphere wasn't even more dominant, but that is a really interesting thing to observe. We have subject, and here this is subject to left minus right homologous pairs, MEG channel M1 viewed laterally. And in this visualization, the left hemisphere does appear to be higher in activity in the lower frequency bands during REM than the right hemisphere, and this is consistent with previous studies. Here we have subject 4, left minus right homologous pairs for MEG channel M1 viewed from the front. And in this visualization, stage 4, or the right side appears to be dominant during stage 4 sleep in the lower frequencies, which is also consistent with previous studies. And one thing worth noting is this is the same data set as what we just saw in the previous slide, but it's viewed from the back. If we shuffle back to the front view and then back to the back, we can see there's significant difference just between the front and back of the brain. So there's so much rich activity going on here that is just waiting to be explored, but I think I'm running out of time here. So with this, I will end by making the point that if you're like me, you'll want to view multiple visualizations of these at once by applying the principle of small multiples and even taking that even further. Here we've got just 16 of these visualizations, and the entire data set has 128 of these that we've generated from the 4 different views for the 4 different subjects and the different channels. So I'd love to take the time to go through them in detail and explore their richness with you, but I think I'm out of time. So with that, I want to once again express my gratitude to ECSS and Mona and David for giving me the chance to work with this amazing data set. And it's been a great experience, and I hope it continues. Thanks very much.
Thank you, Jeff, Mona, and David. Really interesting talk and lots of things happening there. I want to open it up to some questions. I believe I saw one question in chat, at least. Let me see. From Amit to Jeff, how do you pick ISO value to plot ISO surfaces? Can generate artificial artifacts due to interpolation. Do they impact analysis in this case?
Right. Yeah. Not only is that I'm hearing an echo. But yeah, I mean, thanks for that question. Not only is that an issue, there's a lot of things that I would love to discuss about the constraints and all of the issues that have to be brought to bear with these kinds of visualizations. So yes, it's true, isosurfaces through their interpolation can generate artifacts. The extent to which this is a problem, I think, is still up for debate. You know, just the proof is in the pudding to some extent. I mean, the visualizations seem to, in some ways, the activity correlates very well with the sleep stages, but not entirely. And it's hard to say if that's the data or if that's the technician, the expert who rated the data. So there's a lot yet to be sorted out with that as well. I'd love your feedback on that as well.
Great. We have a few more minutes for questions. Are there other questions for either Jeff or on the visualization, or Mona on the data analysis? Lots of data processing, as Jeff and Mona indicated, and or for David on the science and the subject matter here.
Yeah, there's another question there from Ellen. Did David learn something new from the viz?
Definitely. I mean, you know, because our previous studies in the works of others was just a single homologous pair, we see how much variance there is now in the entire brain. And that's why you know, we had, at one point, we wanted to compare each individual homologous pair against all the others, but so far we've only, of course, looked at the whole head, or the front head versus the back of the head versus the whole head, or the four regions of temporal, parietal, occipital, and frontal regions. So we see the differences in it. It's not unexpected that there would be this kind of complexity in the data. So it only enriches things, but the basic hypothesis and basic finding still shows that the two hemispheres of the brain alternate in dominance. We've known that for a long time during waking and sleep, and not too many people are paying attention to it. You know, the ultradian rhythm of how the two hemispheres of the brain alternate in dominance regulates all of your emotions, your performance efficiencies on a variety of tasks, your desires, your drive, everything under the sun, really. So it's a very important rhythm. I hope that helps answer your question.
Yeah, great question and great response. Other questions from the group? Let me just ask real quickly then, David, so you mentioned just now all the things that this can affect. Thoughts on how you would take what you find here and apply it, any particular applications that you're interested in?
Well, you know, this is just what we're looking at here is just it's real basic neuroscience, right?
Yeah.
And of course, what it helps to do is it brings about more awareness and attention to this ultradian rhythm. You can imagine in 1983, we showed how the nasal cycle and the two hemispheres alternate in dominance and phase with each other. And only a little bit of attention has been given to that. So in a way, this helps to expand the big field of psychophysiology in ways that are not molecular per se. You know, a lot of things have drifted off into the realm of molecular biology alone. And so this is getting back to the whole body, mind-body physiology.
It's fantastic. So, yep, that's wonderful. And a lot of detail for people to then look at and find applications for, I'm sure, from the basic science here. So any other questions? We're about at our hour here. Any final questions for our group?
So is this data shared and, you know, maybe viewable by others? I mean, how do you go about, I mean, if and when you want to share this to the community, how is it going to happen?
Well, when I got the raw data from Eric Hallgren and Nima Degani, they said, don't give it out to others. And I'm sort of respecting their you know, priority because it is a super rare data set. You know, it was recorded originally in about 2006 at Massachusetts General Hospital. I have to abide by their rules with me. And so it's not that we can just put up the raw data on the web. You know, I regret that, but I hope that answers your question. Yeah.
What about the derived data? I mean, the raw data probably still, you know, under some kind of a.
Well, we haven't published it yet, for one thing. So we're reluctant to just, you know, splatter it into the world, so to speak, without getting some, you know, reward.
Maybe after publication, you probably provide this.
Well, we'll see, because we have a number of publications in mind with this data. And so to just throw it out there is, you know, there's two sides of that coin, right?
Sure. I mean, of course, you need to publish, and then maybe after that, maybe there are other things to learn from this by other community, you know, in the community, right?
Yeah, well, we're happy to collaborate with people downstream, you know, if it's appropriate from their end and our end. If somebody can bring something to it, but I don't want to sound like I'm limiting the data, but there's certain criteria that that play in here. I hope that answers your question in a fair way.
Thanks.
Yep. Thank you, David. Thanks, Jeff and Mono. Great presentation. Really interesting. Appreciate the time you shared with us and everyone for joining in. And that's it for us today. And hope everyone joins us again for the next symposium.
Thank you.
Thank you all.
Thanks,
Thank you.
everyone.
Bye-bye.
Thank you.
Bye-bye.